Hi, my name is Berk Alpay. I'm a fourth-year undergraduate at the University of Connecticut. I'm going to be talking about two models we've developed for predicting gene expression from genotype. I'm going to start by explaining why gene expression prediction is important. So for many years now, researchers have been doing these genome-wide association studies that try to find direct statistical associations between genetic variants and phenotypes. Since these studies deal with a very large number of genetic variants, there's a heavy multiple hypothesis testing burden. A lot of the apparent associations are by chance. So you can correct for this, but your statistical power to detect true associations is low. Transcriptome-wide association studies alleviate this burden by performing fewer tests. Right, you're dealing with transcripts, not variants, but they lose the connection to genotype. So you'll notice there's this missing component of associating genotype with the transcriptome. If we could fill it in other words, accurately impute expression from genotype, something like a predictive and genome-wide counterpart of cis-eQTL studies, we could then associate the transcriptome with phenotype. We'd have the best of both worlds. So the statistical power of TWAS with the genetic associations revealed by GWAS. So with the availability of genotype and corresponding expression data, there have been some models that try to fill in that gap. Namely, Manor and Segal performed KNN and elastic net regression on minor allele counts or dosages of cis-SNPs, and they did this for each gene against normalized expression. If you're unfamiliar with elastic net, I'll be talking about a lot in this presentation. It's basically just a linear regression model, but with L1 and L2 penalties. The prediction expression model was very similar to Manor and Segal's elastic net, but this was actually the first model to perform association with phenotypes. So soon after that, this model, Slinger, extended the features in prediction from cis-SNPs to SNPs genome-wide. So it's important to note that each of these methods are actually training a model for each gene. Some of these models can be quite accurate in predicting the expression of some genes, but they have low or near zero accuracy for many others. Now, we know epistatic interactions are involved in phenotypes. Maybe methods that selectively incorporate epistatic effects would better model some genes, as opposed to dosage additive models such as PredictScan. The idea of the methods we've developed is to generate a set of candidate tracks. So tracks are haplotypes that start and end at fixed positions. And once we have a set of candidate tracks, to find some tracks among those that are indicative of expression. So first I'll define more formally what exactly it is I mean by the expression regression problem, and present one of the methods, HaplexR, that we've developed to address it. And after that, I'll introduce a new problem, the expression classification problem, and present our second method, HaplexD, tailored to that. What is the expression regression problem? We can describe it generally as attempting to map a haplotype matrix with an individual's haplotypes in the rows and variants in the columns to a vector of real valued expression with an entry for each individual. We take a more specific view of the problem in our paper in that we deal with cis variants of each gene whose expression we're trying to predict. In practice, we assume the infinite sites model of genetic variation and encode each variant in a haplotype using binary indicators. In fact, this is a generalization of the minor allele count representation used by current methods, and this is a generalization to haplotypes, right? So I talked earlier about finding signature tracks. Well, first we need to generate candidate tracks. Namely, we start the first position in the cis window of the gene, and proceed along the genome until the number of unique haplotypes from I to the current position exceed a marginal partition size, which we call big J. So let's look at a toy example with marginal partition size 3, with 3 individuals and their respective haplotypes across 7 variants. We start at the first position, and so far there are 2 unique haplotypes, 0 and 1. The number of unique haplotypes isn't greater than 3, which is big J, so we proceed to the next position. Now there are 3 unique haplotypes, 0, 1, 0, 0, and 1, 1. We still haven't exceeded 3 unique haplotypes, so we go on. We still haven't exceeded 3, still haven't exceeded 3, and now by the 5th position, we have 4 unique haplotypes. So we've exceeded the marginal partition size. And now we label the unique haplotypes in the first block with cluster IDs and start a new block, right? So we reach the end of the sys window without exceeding big J. So we end the block, label the unique haplotypes, and that's the end of the clustering algorithm, essentially. So now, if we wanted to visualize these clusters of a graph, it would look something like this, right? If we pass a haplotype through this graph, we'd get a cluster ID at each block. So how do we actually encode these clusterings in our model? Well, we have our cluster IDs, and we one-hot encode each haplotype in each block, and sum the 2 one-hot encodings for each individual. So if we look at the second block for donor A, we see her haplotypes are both in cluster 1, which is encoded as the vector 0, 0. So the sum of the haplotype encodings is 0, 0. One of donor B's haplotypes is in cluster 2, which is encoded as 1, 0, and her other haplotype is in cluster 3, which is encoded as 0, 1. We add them and we have the vector 1, 1 as her encoded cluster memberships. And we can proceed similarly for the others. So now that we have our clusterings, how do we actually make predictions about expression? The first part of the input to the model is actually identical to the input to PredictScan, namely minor allele counts in a sys window of the gene. But then we append to this design matrix the haplotype cluster memberships. So this is what we derived in the previous slide. And in fact, if we look at these 2 entries, we'll notice that one of individual 1's haplotypes is in cluster 1 of the first block, and the other is in cluster 3. So, this is our design matrix. And then we train elastic net regression against the normalized expression of each individual. The vector of these expression values across individuals is what we call y. And in this equation, x sub d is the design matrix, the betas are linear regression coefficients, and lambda specify the penalties on the betas. And in practice, we perform cross-validation to select the lambda parameters. To validate our model, we use 5 tissues with relatively high sample counts from version 8 of GTEx. We pre-process to select common biallelic SNPs. We do LD pruning, normalize the expression, and correct it for genotype PCs, peer factors, etc. So from this data, we select 15,000 genes and a 10% holdout of donors on which to evaluate some models. So we compared the performance of haplex R on these genes across all tissues that we selected against the KNN and PredictScan models I discussed in the related works, along with LASSO, which is a dosage additive model like PredictScan, but it uses a different penalty on the regression coefficients. We noticed that our model performs better than the dosage additive models for about half of all genes, and more so against KNN. So in fact, we noticed there's substantial subsets of genes for which haplex R is the best model, and for which it's the only model that passes certain thresholds of accuracy. This indicates that haplex R captures non-additive signals of variant effects on expression, and that these signals are generally of higher quality than those found by KNN. To investigate these non-additive signals, we did a grammar analysis in GCTA to estimate the variant additive heritability of each gene. So in the left figure, the blue points are the narrow sense heritability of each gene in increasing order, and the red points are the model R-squareds. Essentially, we found that haplex R detects more non-additive components of heritability than the dosage additive models, PredictScan and LASSO. The predictive accuracy of KNN is greater than the narrow sense heritability of a larger proportion of genes than what we see in haplex R. But when we look at the lower tail of the KNN figure, these models seem to be of lower quality. We also found that haplex R variants at beta thresholds were enriched in various regulatory annotations across tissues. We noticed enrichments when we restricted the variant sets to contain SNPs, haplotypes, and both. So now let's move on to predicting discrete expression. The problem formulation is very similar to the expression regression problem, except that we now want to predict classes of expression. Right? So why might we want to do this? What's the biological motivation? So low and high expression groups of some genes appear to be indicative of risk for some diseases. The left figure is from a paper on Alzheimer's disease, and the right is a paper on colorectal cancer. So also, RNA transcript reads are not guaranteed to be normally distributed, right? In fact, the distribution of reads may have several modes which are important to capture in expression prediction. For discrete expression prediction, we take a more combinatorial approach to finding signature tracks. A suffix tree is a data structure in general that encodes substring sharing. In our paper, we use a modified version of a suffix tree that, instead of encoding just haplotypes, encodes the positions of the haplotypes in addition to their allelic values. The end result is a trackized suffix tree. So this is a suffix tree that encodes track sharing. Let's look at a dummy example. Here are three haplotypes, X0, X1, and X2, and their corresponding trackized encodings. Let's insert them into a suffix tree. So here the dollar signs are a special character indicating the end of a haplotype. And from this we can notice that each root to node path is actually a tract. So we have our candidate tracks, but how do we select those that are informative with respect to expression? So first we define this idea of tree clusters. These are the two sets of haplotypes diverging from a non-root internal node. We rank these tree clusters by their ability to separate expression classes. Here's a toy example with contingency tables for a highly informative tree cluster, this is Z5, a not so informative tree cluster, Z sub 100, and an unformative tree cluster, Z sub 200. We can confirm this visually, but in practice we use a chi-square test and a conditional entropy to evaluate the clusters. So far we've generated candidate tracks and ranked them, but how do we generate predictions about expression? Well, we build an adjacency matrix between the haplotypes that encodes their genetic similarities with regard to the top T informative tracts. And then on top of that, this is a dummy example, on top of that we can perform a spectral clustering on this weighted graph to obtain clusters of genetically similar haplotypes. We predict the expression of a haplotype as the majority expression class of its cluster in this graph. An individual's haplotypes may fall in different clusters, in which case we predict the class indicated by the cluster with highest purity. Now that we're moving into the results, I should note that expression data isn't given to us in a discrete and covariate corrected form. Instead, we take the normalized and covariate corrected expression and we discretize it using the principle of maximum entropy. So we validated haplex D on three settings of the number of expression classes, E equals 2, E equals 3, and E equals 4. And we compared them against the regression models I presented earlier. Since we're now looking at classification, we discretize the predictions of these regression models according to the bins of the maximum entropy partition. We also compared our models against logistic regression with ElasticNet and lasso penalties. The takeaway is that haplex D shows significantly higher classification accuracy in each case. And when we look at the F1 scores for each method and tissue, we actually notice that the performance of haplex D relative to the other models increases with the number of expression classes. So in sum, in our work, we introduce the gene expression classification problem and present expression models that relax the assumptions made by purely dosage additive models. There are some limitations to our methods. Namely, we require phased genotype data. And when we look at our algorithms for generating candidate tracks, we see that they're not really robust to phasing and variant calling. In both haplex D and haplex R, haplotype can easily fall out of a cluster due to a change at a single locus. So this goes to show that there's a large space of genetic models that we haven't explored, and that some may be custom suited to detecting genetic patterns with respect to expression. We focus only on cis-variant effects, but in the future we may also wish to model regulatory markers and transvariants. I want to take a second to thank my co-authors on this work, in particular Pinar for leading the development of haplex D, and Professors Israel and Aguirre for supervising this work. And thank you all for your attention.